In the ongoing lecture series on plant tissue culture, in my previous lecture I started with hairy root cultures. In that we discussed what are hairy roots and how the infection caused by bacteria rhizobium rhizogenes get transferred into the plant genome, how the DNA from the bacteria gets transferred into the plant genome, thereby leading to the infection which we call it as hairy root. Or in simple words, we can say that hairy root is nothing but it's a pathological condition for the plants that is caused by the bacteria rhizobium rhizogenes. Then we discuss about the factors that affect or the problems that occur when we are scaling up this hairy root culture up to a commercial level or in bioreactors. We also discuss about various factors including the shear stress caused by the impellers, proper mixing of the culture media, then proper transformation of the mass, nutrient mass or the oxygen circulation, and so on and so forth. We discuss all the factors that can impact the growth of hairy root under the culture conditions. We also discuss about the methods by which we can assess the growth of the culture under artificial conditions, and in that we discuss about various softwares that are being used nowadays for detecting the growth of the roots in terms of its length, branching points, root tips, and so on and so forth. We also discuss about the genetic algorithms and artificial neural networks that are used in order to assess or in order to optimize the cultural conditions for proper growth of hairy roots. So friends, in continuation with the previous lecture, today we are going to start with the applications, which is nothing but the last part of hairy root culture lectures. Now as we can see that hairy root culture has a wide number of applications ranging from phytoremediation, then production of secondary metabolite, biotransformations of the secondary metabolites, molecular farming. It also helps in germplasm preservation, then in production of recombinant proteins, also in understanding the host pathogen interactions. Hairy root cultures are also used for polyploidy alterations, then to produce stress tolerant species, and in order to understand the biosynthetic pathway or to elucidate the biosynthetic pathway in the plants. Now if we broadly categorize all these applications, then we can sum up all the applications in 4 major headings, that is phytoremediations, the production of secondary metabolites, biotransformation processes, and molecular farming, because these are the 4 major applications of hairy root culture. So coming on to the use of hairy root cultures in secondary metabolite production. Now in previous lecture also we discussed certain advantages of hairy root, that is they have a very fast growth rate in comparison to the conventional roots, and therefore they require very less time to grow to a substantial amount. Similarly, they have a very low doubling time. When we say doubling time, it means they get the volume of hairy roots get double in a very short period in comparison to the conventional roots. Further, these roots also have an advantage of ease of maintenance. Once you have optimized the culture conditions, once you have optimized the temperature and proper temperature and pH conditions, you have optimized the viscosity of the media, you have optimized the speed of the impellers which are used to introduce rotation for the proper mass transfer and oxygen circulation in the media. Once all these parameters have been optimized, these hairy roots are easy to maintain. Then they have an ability to synthesize variety of phytocompounds, ranging from simplest compounds to more complex compounds. We can use hairy roots to synthesize variety of phytocompounds, and not only they are used to synthesize variety of phytocompounds, but they have an ability to synthesize and store more amount of phytoconstituents in comparison to conventional roots. Now since they have more ability to synthesize and store phytoconstituents in comparison to the conventional cells or conventional roots, they become more economical for the commercial scale production. Now, by using hairy root culture, we can enhance the secondary metabolite production, and this enhancement or increase in the production of secondary metabolites can be done by using various strategies. And the first one is by adding the precursor. Now, precursor is a molecule that leads to the biosynthesis of the desired compound into the plant matrix or into the plant cell system. Now, there are certain disadvantages of adding the precursor into the culture media, as it can be a costly approach, because we know that precursors are normally synthesized in the plant system, and if it is very difficult to isolate that precursor, or that precursor is being synthesized in the plant in a very small amount, that precursor can be very costly, and therefore that the whole process will become costly. Second is, the direct precursor may not be available from natural sources. So, as I told you that certain plants or in some plants, the precursor may be synthesized in a very small amount, or you may not be able to isolate that precursor that can be used for the commercial scale. So, you may not be able to isolate the precursor in large amounts so that it can be used for commercial scale. Therefore, that the process again becomes more costly. And third is, the chemical synthesis of the precursor or the chemical synthesis process of that precursor is not known or is difficult to synthesize. And if this happens, again we will be dependent upon the precursor from the natural sources, and again the process will become very costly. So, another strategy is, we can increase the production of the precursor. Say for example, if the precursor is being synthesized in a very small amount in the plant matrix, we can devise a mechanism to enhance the production of that precursor, which leads to the enhanced production of the desired secondary metabolite. And how it can be done? I have taken an example. It was observed that production of podophyllatoxin, which is a very well known anti-cancer lignan compound, by Podophyllum hexandrum cell suspensions was limited due to the availability of coniferin, which act as a precursor for podophyllatoxin. So, in order to overcome this limitation, in order to overcome this problem, Podophyllum hexandrum cells were cultured along with the cells of Linum flavum in hairy root culture, and this culturing of Podophyllum hexandrum cells along with Linum flavum enhances the production of coniferin in the culture, which eventually increases the production of podophyllatoxin by 240%. So, this is how we can increase the production of precursor in the culture cell by culturing the desired cells with the with another type of cross species cells, which can lead to the enhancement in the production of the precursor molecule. We can also go for metabolic engineering or genetic modification in the plant cells through hairy root cultures, by which we can enhance the production of secondary metabolites. Now, I have taken an example of the alkaloids that are synthesized by Catharanthus roseus, also known as Vinca rosea, and some of the solanaceous plants. Now, these alkaloids are synthesized through tryptophan or tryptamine, and it has been found that the levels of tryptophan and tryptamine, they become the limiting factor for the production of these alkaloids. Now, why this happens? Because tryptamine follows a negative feedback mechanism, which basically inhibits the synthesis of the desirable alkaloids. So, as soon as tryptamine is synthesized in the plant cells, it sends the negative feedback signals to the other cells, which stop the production of tryptamine and tryptophan into the plant cells, and until the tryptamine, which has been biosynthesized, is completely transferred into the alkaloids, then that negative feedback mechanism stays in place. As soon as the whole of the tryptamine is consumed, this negative feedback gets nullified and fresh tryptamine is synthesized. So, that is why we said that tryptophan and tryptamine are nothing but they are the limiting factors in the production of alkaloids. Now, what happens exactly is, tryptamine is produced from tryptophan by the action of tryptophan decarboxylase enzyme. And tryptophan is produced by anthranilate synthase enzyme in the shikimic acid pathway. So what we can do is we introduce a gene of Arabidopsis thaliana which encodes or which upregulates the activity of anthranilate synthase enzyme. Or in other words, we can say that it makes this particular enzyme insensitive to the negative feedback mechanism. So what happens exactly is, so as soon as this enzyme gets insensitive or it gets upregulated or it becomes non-responsive to the negative feedback mechanism, so as soon as that tryptamine is produced, this enzyme does not get deactivated. So as soon as anthranilate synthase does not get deactivated, it keeps on producing tryptophan, and tryptophan by action of tryptophan decarboxylate will continuously producing the tryptamine, which will eventually enhance the overall synthesis of the desired alkaloids. So this was one example by which you can genetically modify the plant using hairy root cultures. Another example is solanoside glycoside, which is again an anti-cancer agent produced by Solanum khasianum plant, and it negatively control its own biosynthesis through negative feedback mechanism. So as soon as solanoside glycoside is produced, it sends negative feedback signals to the biosynthetic pathway and it stops producing the solanoside glycosides. Now to overcome again this negative feedback mechanism, a recombinant antibody which was expressed from a foreign gene was taken and it was transferred into the hairy root genome. Now this gene or a protein, it bind to solanoside glycoside, thereby inhibiting it to initiate the negative feedback signals. Once this negative feedback signals were inhibited, the production of solanoside or the biosynthesis of solanoside glycoside was resumed, and by this mechanism the overall production of solanoside glycoside was enhanced 2 to 3 times in the cultured cells. Another approach by which we can enhance the secondary metabolite production is by using the metabolite traps. Now what is a metabolite trap? It is a sequestration and recovering process which is achieved by incorporating a resin or an organic phase which is used to increase the production of metabolite or to avoid their degradation in the culture media. Or in simple words, I will say that it is nothing but it is a process by which we trap the desired secondary metabolites to enhance their production and to avoid their degradation in the culture media. But the problem with metabolite traps is that the usage of a particular metabolite trap will depend upon the chemical nature of the secondary metabolite, because for trapping we need to have certain chemical interactions or binding of the secondary metabolites with those trap molecules, and therefore the usage of metabolite trap largely depends upon the chemical nature of the secondary metabolite that is being produced in the hairy root culture. Now there was a study on beta vulgaris wherein there a combination of alumina and silica in ratio of 1:1 was added to the culture media. Now by addition of this alumina silica combination, which act as a metabolite trap, it enhances the recovery of the colored pigments, that is betalains, by 97%. So this is how this technique helps in trapping the desired secondary metabolites and enhancing the recovery of these metabolites from the cultured media. In another study, a hydrophobic resin X5 was added to the media, and by addition of this X5 resin, which again act as a metabolite trap, there was an increased production as well as increased recovery of the tanchinones from Salvia miltorrhiza plant. So both production as well as the recovery of the desired secondary metabolite increases significantly by using these metabolite traps. Another approach by which we can enhance the secondary metabolite production is by using the elicitors. Now elicitors are the agents that stimulate the production of secondary metabolites, and depending upon the nature of the elicitors, these can be biotic or abiotic elicitors. Now, biotic obviously means they are the organisms, which can be a bacteria, it can be a fungus, it can be a virus, whereas abiotic elicitors include usage of high temperature conditions or usage of osmotic stress or pH conditions and so on and so forth. So, we can use elicitor agents or eliciting agents to enhance the production of secondary metabolites into the hairy root cultures. Now, there were certain studies regarding the usage of elicitors to enhance the production, and one was usage of osmotic stress by high sorbitol concentration along with a yeast elicitor. So, here yeast acted as a biotic elicitor, whereas osmotic stress or high sorbitol concentration acted as a abiotic elicitor. Now, usage of combination of abiotic and biotic elicitors in case of Salvia miltioryza enhances the production of tanchinone by almost 100 folds in the hairy root cultures. Another example was using a sublethal intensity of electric current. So, again electric current here acted as an abiotic elicitor in Pisum sativum or the pea plant, which enhances the production of picetin, which is the active constituent, significantly in hairy root cultures of pea plant. Then methyl jasmonate, which is nothing but a stress hormone in the plants, is a biotic elicitor. It enhances the ginsenoside production in hairy root cultures of Panax ginseng, and we know that ginsenosides are the active constituents of the Panax ginseng, or the majority of the pharmacological activities exhibited by ginseng are because of these compounds, ginsenosides. Now, another application of hairy root culture is the biotransformations. So, if I talk about biotransformation in simple words, it is nothing but it is a process of conversion of a particular molecule into a molecule which is desired. So, exact definition if we talk about of biotransformation is, this is a specific modification of a given compound to a distinct and structurally similar product by a biological system, which could be microbes, animals or higher plants. So, what we do in biotransformation is, we introduce a specific enzyme or a bacteria which contains that enzyme, and that enzyme acts upon a specific precursor molecule or an isomer of the desired molecule, thereby changing its stereochemistry to the desired molecule. Say for example, if a plant produces L isomer of the desired compound, but it has been found that the D isomer of that compound is more active, then we can introduce certain enzymes in the culture which can convert that L isomer into D isomer, and we can obtain higher concentration of D isomers which are more pharmacologically active. Now, during the biotransformation processes, different types of reactions are involved, such as oxidation is there, glycosylation is there, reduction reactions are there, hydroxylation reactions are there. So, numerous types of reactions are involved in this process in the culture, which leads to the changes in the exogenously applied or endogenously produced substrate. So, if you are adding a specific precursor molecule from outside, which we discuss in case of enhancement of secondary metabolites, then also these biotransformation can act on those exogenously applied molecules or maybe precursors to enhance the production of the desired secondary metabolites. Or if that precursor or a molecule is produced inside the plant matrix through biosynthetic pathway, that can also be changed by using the biotransformating agents. Now, biotransformation can be done by feeding the precursor molecule, which we just discussed, that we can introduce the precursor molecule from outside, and by maintaining the biotransformation conditions in the culture, we can enhance the production of the desired molecule. Say for example, hyoscyamine if added in the cultures of Hyoscyamus niger, that enhances the production of scopolamine. Now, how hyoscyamine gets converted into scopolamine is that hyoscyamine undergo epoxidation reactions to give rise to scopolamine, which is also known as hyoscine. So, if we increase the production of hyoscyamine in the culture and we introduce that specific enzyme which results in epoxidation of hyoscyamine, we can enhance the production of scopolamine or hyoscine in those culture of Hyoscyamus niger. Another example is It was found that feeding of amino acids, specifically phenylalanine and cysteine, in Tropelium majus plant hairy root culture, it enhances the production of glucotropiolin, which shows that the amino acid precursor availability was the limiting factor for the production of glucotropiolin molecule. So, this also helps in understanding what is the biosynthetic pathway of a desired secondary metabolite, that what are the major molecules that can act as the limiting factor for the production of this secondary metabolite, so that we can exogenously add those limiting factors, we can exogenously add the microbes or the viruses which or the fungi which can produce those enzymes which convert those molecules into the desired molecules. Then we can also upregulate the enzymes responsible for the conversion of precursor to the desired compound. Now, it has been seen that the upregulation of hyoscyamine 6 beta hydroxylase gene in hairy root cultures of Nicotiana tabacum resulted in bioconversion of exogenously supplied hyoscyamine to more scopolamine. So, another example, we add more amount of enzyme into the culture by which can convert hyoscyamine into hyoscine. Then biotransformation can also be done by using co-culture techniques. So, this is another strategy by which we can go for co-culturing of plant cells, we can go for co-culturing using same species, we can go for co-culturing using different species. So, in case of using same species co-culture, it was found that whenever Atropa belladonna shooty tetromas and hairy root were co-cultured, there was a significant increase in the scopolamine production. Now, what happened was, during the hairy root culture, the hyoscyamine that was produced in the hairy root culture, this was transferred or translocated from the hairy root cells into the shoot cells. Now, shooty tetromas are nothing but these are the tumor like outgrowths, tumor like infections caused by the bacteria in the plants. So, once this hyoscyamine was translocated into these tetromas, there the tetroma cells introduce the transformation of hyoscyamine into scopolamine, which thereby enhancing the overall production of scopolamine in the co-culture studies. Then cross species co-culture, we have already discussed this example. The podophyllotoxin content was significantly increased when Linum flavum hairy roots were co-cultured with Podophyllum hexandrum. We can also enhance the biotransformation by adding suitable elicitors like Bacillus cereus. When added to Salvia miltorrhiza hairy root culture, it enhances the production of tashninone. Then another application of hairy root culture is phytoremediation. Now, phytoremediation can be done by phytoextraction, phytovolatilization, phytostabilization, phytostimulation and phytodegradation. Extraction is nothing but to extract the toxic residues or metals or pesticides or pollutants from the soil, and they get accumulated in the plant cells. Volatilization, once they are extracted by the plant, they are volatilized into the environment. Stabilizing, they bind to certain proteins, making them non-degradable into the soil. And by phytostimulation is enhancing the activity of the enzymes which degrade those pollutants within the soil and in the plant. And another one is the phytodegradation. Now, for such purpose, hyperaccumulator plant species are used, like Alyssum species, Sedum alfaredi is there, Brassica junicea is there, which are hyperaccumulator plants for copper, zinc, nickel, cadmium and so on and so forth. Another example is Cucurbita pepo or Raphanus sativus is there, which is used for phytoremediation of various pesticides. PCBs are there, HCH are there, DTD is there and so on and so forth. But the problem is these pollutants are hydrophobic in nature. So, what we can do is we can genetically modify the plant cells to enhance their absorption, and by which we can remove the toxicants from the soil. We can also enhance the activity of certain enzymes which can degrade those pollutants that are present in the soil. So, friends, this was all about hairy root cultures, and in the next lecture we are going to start with another technique, that is organogenesis.